Welcome to the Heart of CIRS podcast. I'm your hostess, Melanie Joy Pensack, here to share heartfelt conversations with folks recovering from CIRS and with those special people serving the CIRS community. The podcast was created to help bring awareness to the physical, emotional, and mental experiences of folks navigating CIRS day to day. The world needs to know what CIRS folks go through for deeper empathy and understanding. Through stories and vulnerability, we can help the world understand the winding journey of CIRS recovery. Thank you for being here to open your mind and to open your heart. I'm very much looking forward to being in conversation today with GENIE Ryan on the podcast. Looking forward to diving in to help people understand a little bit more about the GENIE test, which, as some of you in the CIRS world may know, it's a more advanced blood test used by many of the CIRS docs to get more detailed information during diagnosis. And GENIE stands for Gene Expression Inflammation Explained. I'd like to introduce GENIE Ryan to you today, who will be here, who is one of the co-creators of the GENIE test. GENIE Ryan is a molecular biologist who lives in Vero Beach, Florida. He graduated from Florida Atlantic University with a BS in biology, BA in chemistry, and a BA in physics, all with honors. He graduated with a PhD in the Department of Neuroscience and Physiology from the Medical University of South Carolina in 2001. And followed that with a postdoc appointment at the University of Miami Miller School of Medicine. He has published over 30 peer-reviewed papers, mostly in the field of genomics, and 2 book chapters. In 2003, he began working in the Marine Biotoxins Program, National Oceanic and Atmospheric Administration, where for 8 years he studied gene expression in cell culture and mouse models after exposure to marine algal toxins. Thank you so much for being here today, Dr. Ryan. We're really grateful for your time and looking forward to diving into this conversation.
My pleasure.
So before we get into some of the GENIE details, part of my podcast is helping people get to know the people behind the CIRS world pieces. And so I'd love for people to learn a bit more about you personally. And I'm curious, how did you get into science? Were you interested in the body as a kid, and what led you to your degree?
Well, I guess I'm probably not going to be the answer most people would expect, but I was a very average high school student. I was smart, but I think a lot of kids, sometimes high schools, you're there for different reasons, your social interactions and fun, and that's why I was there. So my academic prowess was not very good in high school, made it out. And after high school, I just started working and did some construction work for a year and decided that was too hard. And then I followed the sun. I grew up in Maryland. And when I was young in Maryland, I was considered that to be the North and didn't like the cold weather. So I moved to Florida. And I bounced around for 5 years. I moved down to Florida in the winter and then moved back up to Maryland in the summer. And like I said, did that for 5 years, had a lot of fun. And I guess at that point, I realized I really wasn't going anywhere. So I thought maybe I should go to college. And I started at a community college and just took freshman classes. And I did like biology, so maybe I gravitated towards the sciences immediately. I was always pretty good in math. And so when I started college, I ended up liking the science classes that I was in and decided to major in biology. And I got to the end of my major in biology and was left unsatisfied with a lot of the answers you get in biology as to how things work. The answer is, why does your heart speed up when you're scared? Well, that's adrenaline. And there's always another follow-up question. How does adrenaline work? A molecule that binds to a receptor. Pretty unsatisfying answer. So that led me into chemistry to actually understand how molecules interact and again, a little unsatisfying, which then led me into physics to understand more how electric fields work and the physics of how everything interacts. And I guess at that point, I finally had enough schooling, and I was satisfied now with my understanding of how things work. And I was left with 3 degrees in science and decided to pursue a PhD in a molecular science so I could use my physics and chemistry background. And so I really drifted through, but gravitated towards the things I was curious about. And I guess that's a luxury, just to be able to follow your curiosity. And that just led me into molecular biology, which is the study of how biology works on a molecular level. By the time I finished my PhD and went on to do postgraduate work, I was just fascinated with how physiology works and where it comes from. Where does it begin? And if you find yourself asking that question, where do things begin in regards of physiology? They begin at the DNA. Governs your entire physiology. So that's how I ended up studying genomics.
Fascinating. So at what point did you intersect and connect with Dr. Shoemaker's work?
That's also a pretty interesting story. So you had mentioned in my bio that I worked for the National Oceanic and Atmospheric Administration in the Marine Biotoxins Program. And while I was there, I was helping to develop a program in toxicogenomics. And it's funny because at the National Oceanic and Atmospheric Administration, you'd think, well, why do you study toxins at NOAA? It's because there are poisonous algae in the nearshore waters that fish eat, and fish become toxic because they eat the toxic algae. Then people eat the toxic fish and they get sick. And so that's why NOAA studies marine biotoxins, because they're in the oceans, and we want to make sure we know when they make their way into the food chain and into human consumption. So part of my role at NOAA was understanding the effects of these harmful algal toxins on animal physiology, because part of the mission at NOAA is also to study the animals in the ocean. So when you hear about a lot of dolphins washing up for unusual, it's called an unusual mortality event, when a bunch of marine mammals wash up on a beach dead or dying. And it's normally because sometimes they can get disoriented and just wash up, but sometimes they eat fish that are highly toxic and they just become so sick and they just beach themselves. And sometimes you'll see hundreds of marine mammals beaching themselves because they've eaten toxic things. As marine mammals eat these toxic things, so do humans. And one of the toxins they eat is called ciguatoxin. And so I was doing a big research project looking at the effects of ciguatoxins on gene expression in mammals. I was using mice and cell culture to look at genes and how they change, how the expression changes when they're exposed to ciguatoxins. And I graduated really in the Department of Neuroscience and Physiology. So I had a neuroscience background, and these are neurotoxins. And as I'm studying these, what became a little confusing to me was why, when I was looking at the genes responding to these neurotoxins, the overwhelming effect was inflammation. And I was thinking I was going to find more nerve-related injury, but actually I was finding more immune-related activation in response to these injuries. And so I've gone into my boss's office to say, look, I know we're studying neurotoxins, but here's what I'm finding. I'm finding a lot of inflammatory mechanisms are just running out of control when these animals are exposed to these toxins. And I put the summary of results, I handed it to him, and he looked at it and looked it over for 30 seconds or so. And he looked up at me and he goes, I've seen this before. And he opened his desk drawer and he pulled out this piece of paper and he handed it to me. And that piece of paper was Dr. Shoemaker's biotoxin pathway. And so he handed me this piece of paper and he goes, call this guy. And I did. And that was how I met Richie. So that was the crossover where human physiology met animal physiology. And so I've been working with Richie probably now for almost 15 years. We started working in collaboration while I was still at NOAA. So we did some collaborating there on ciguatoxins and human exposure. And then Richie just said, why don't you just come work with me and we'll study people exposed to microbes in water damaged buildings, because there's a lot more of those people getting sick than people eating ciguatoxin. So that's how we started.
Wow. I love how in the CIRS world, there are many different stories of how people have connected and these different synchronicities. It's really interesting to see how the work has come together. What year did you and Richie then develop the GENIE?
The GENIE went through a couple technology changes. So maybe we started working on it a little over 10 years ago. And at the time, we were using a technology called microarray, which was certainly replaced by genome sequencing. And so we started with microarray, but with the advent of newer technologies, we decided to switch over to this next generation sequencing. And so the development took a little bit longer than we hoped, but I think we made the right choice in switching our technology platform. It's hard to find a microarray platform nowadays.
Let's explain to people a bit more about what the GENIE test is, what it isn't. There will be people listening to this podcast who are completely new to CIRS and maybe have heard of the GENIE test as part of the diagnostic process. There will be people who may be listening and curious about this for friends or family members of those they're trying to understand what's going on with their health. And there might be, other functional practitioners and physicians curious about it as well. So what is the GENIE and what is it not?
I'll first tell you what it is not. It is not a test that looks at genes as far as mutations in genes. It does not look at the DNA. It looks at a copy of the DNA called RNA. And I don't want to get into the weeds on all these technical terms, but RNA is an intermediate between the DNA and the final protein product. And the reason we look at the RNA is because it's easy to measure, and we can measure all of it at one time, which is a great advantage when you're dealing with a complex medical illness with very little clues as to what is driving the pathology. And even when I explain this to people who are familiar with DNA and proteins and RNA, it's still very complex. And an analogy that's very helpful sometimes is to think of this process as a musical instrument, let's just say a guitar. So in the body, information flows. The DNA contains all the information of an organism, but the DNA is static. It really can't do anything. It needs a dynamic process to fulfill its mission. And in this analogy, the DNA is like a guitar. It's fixed. The guitar doesn't really change. Now, you can tune a guitar and change it that way, and that would be like epigenetic changes, if people have ever heard about that term, epigenetic modifications. So you can modify the guitar slightly, but the guitar is still basically the same. What is important with a guitar is the music that it plays. So a guitar can't play music by itself. It needs outside intervention, so it needs someone to hit the strings. And that would be the environment. When the environment comes in contact with the DNA, which would be the environment of your cells, the DNA will produce these RNA molecules, and those RNA molecules go out into the cell, and proteins are produced from them, and those proteins do whatever the cell needs to be done based on the environment you're in. And looking at the RNA is like listening to the music coming off the guitar. Everyone basically has the same guitar. We're all humans and we basically have the same instrument, but we're all playing different music. Our focus is not the fixed DNA. It's the response of your DNA to your environment. So it's complex. And people recently, and I would say in the last 6 months, I've just been really thinking about this because people ask me to explain this test. And I would say this is one of the most complex clinical diagnostic tests in the world. This is about as complex as you can get. And I try to leave it at that. But one thing we try to do on our website is give people more information, because sometimes trying to break things down from extremely complex into completely understandable is a long journey.
I've listened to your talks over and over and over again. And it's such a highly complex illness. CIRS is such a highly complex illness. And then the test is so complex as well that it is truly a skill to be able to break this down into language that's accessible for people. So I appreciate your effort. You're doing a great job.
Thank you. But I think it comes back to that curiosity where, like I was studying biology, but biology is incomplete unless you understand chemistry, and chemistry is incomplete unless you understand physics. And you could still go, physics is incomplete unless you understand math. All of these things, if you can bring things down to laws that have to be obeyed, and then you work yourself up from there to try to understand things, that's how the complex can become less complex. There are not many tests that use RNA as a clinical diagnostic because it is extremely fragile. It is one of the most fragile molecules in your body. This is a signal from your DNA, and this signal is meant to be destroyed. It's like a secret message. I'm going to send you this signal and it's going to be gone quickly. So you got to act on this. And there are reasons for that, because you constantly have to change your physiology depending on your environment, depending on lots of things, depending on your diet, depending on your stress levels, depending on your pain levels, depending on what temperature it is outside. Your body is constantly adapting to things in its environment. And Richie was smart enough to recognize that this complex illness is an illness driven by an innate immune activation, which is different from totally different from an adaptive immune activation. So just by figuring that out right off the bat saved a lot of time in discovering all the pathology involved with this illness. So we have somewhat of a focus on what CIRS is that we can look into, but I guess maybe I'll go back. The reason RNA is such a great thing to look at is because I can look at how all the genes are responding in one test, whereas it's just such an advantage that you can take complex systems and analyze the whole system in one test. It's really such a novel medical breakthrough that in the future, I have no doubt you'll be going to the doctor for whatever reason, and they're going to take a blood sample and look at your gene expression and your immune system, and that will be pretty much standard practice going forward. So I think it's really nice to have these tools and be able to apply them pretty much before anyone else can. We got lucky in falling into this relationship where we could take cutting edge scientific technology and apply it to problems of today.
I think this will be a great place for me to jump in and share a little bit about my experience with GENIE, to put what you've just said into what this looks like from a very practical patient perspective. So people will know my story and my significant CIRS journey that started 2001 officially. And I didn't have my first GENIE until 2002 when I got to a CIRS physician. But I think that it will be helpful for me to explain. I'm not going to tell my whole story again because that can be heard in a previous episode, season 1, episode 1, I share a lot about my health journey, but this particular test was groundbreaking for me because by the time I got to my CIRS doctor, I was significantly physically impaired. And there was not a lot of time that I sensed to play around with questionable tests or questionable protocols. It was time to really dig deeply into what was triggering my immune system. And my doctor speaks of this as cheating on the test because essentially CIRS, essentially the GENIE can show you what you are reacting to in the moment, in the time that you get your blood drawn. So for me, being so significantly impaired, it was really important to know what is going on in my body that needs priority for treatment. What is the trigger in my environment that I need to look out for in my next home? And how am I going to remediate or clean myself and clean my body? And at that point, after many years of sort of functional issues, and then CIRS kicked in full force 2021, I had spent so much money. I had so many different diagnoses thrown at me. So when I was in CIRS, but not yet diagnosed with CIRS, people were saying, well, it's Lyme. Well, it sounds like it is Epstein-Barr virus reactivated. It could just be COVID in your system. Even though I wasn't testing positive for any of those things on any of the other blood labs I was having done, people were still throwing those labels at me. And yet I was getting sicker and sicker to the point where I could barely move my body. And so my physician basically said, this has to be done because we need to give you specific care so that we know exactly the best way forward and we can rule in and rule out these labels. And so the first GENIE that I had done helped to explain my chronic fatigue, why I was so weak. It helped to explain which toxins were affecting me, which were actinomyces, the bacteria often found with molds and fungi and bacterias and things, and endotoxins. So for me, I wasn't reacting to mold, even though I tested my environment for mold. And it helped us to know, okay, when you're looking for this next home, you need to really make sure you're looking at the actinos and endotoxins as well. And you need to be cleaning your body in a certain way. And we know that CIRS affects many different systems in the body. And so we're not just wanting to know what's happening with what biotoxin is triggering us. We want to know how else is the whole system affected. So there's a history of blood clots in my family, for example. And so having the coagulation information that you get back from GENIE was really helpful to understand what was happening in that perspective. It helps you to understand how your T cells and B cells are functioning. It helps with explaining your metabolism in different ways. And so this one test really helped to rule in, rule out, and give us a very clear path forward. And what I want to express to folks that are listening to this and on the fence is that you might have had these labels thrown at you as well, but many of those things cost a lot of money and time. So you might try different supplement protocols for Epstein-Barr or for Lyme's that's suspected, that's not showing up on a test, right? And that's hundreds of dollars and many months waiting to see if these supplements and these protocols might work. And certainly some people with CIRS are more functional than others, but there is a significant amount of people that end up with CIRS that are very impaired from a fatigue perspective and a physical perspective. And I fall in that camp. And there wasn't the time to play around with experimenting with all of that. And so, I know people look at the cost of this test, which I think it's around $750 at this point of recording. And then it costs money for the dry ice and to ship it to the lab. So you're talking about $1,000 realistically for this test. But when you look at the big picture of costs of time with doctors and questionable tests and protocols and things that may or may not work, when you do a GENIE, you're getting concise, specific information that saves you time and ultimately saves you money, in my perspective. And so I want to sort of just plant that in there because I think you've really laid out how important and advanced this test actually is. And I don't know that people fully understand and grasp that this is really state of the art to be able to get from 1 or 2 blood samples, all of this information at one time. I mean, that is significant to be able to cheat on the test, per se, as my doctor says.
I'm glad to hear you've gotten a lot of help from the test. And I think this goes back to, even if you get a blood test that says, okay, your TGF-beta is high, and then the next question is, why is it high? So you're always, until you can track things back to the source, you're never going to have a complete answer. But when you can get to the source, you go, oh, okay, now I can see everything. Now there's so many doors that have been opened to look at why, what's driving this. And you can see the drivers of the pathology rather than just the end point of the pathology.
I think we've touched on this a little bit, but is there anything else that you want to say around the process of blood collection? When people decide, okay, or their doctor says, it's time for us to do this test to help give us this specific information and give us a path forward, and they get the test instructions, they are very detailed.
Yeah.
And it is a bit of a process. And so that can feel discouraging to people who have a lot of brain fog and who are working through a lot physically. It is really important, though, that it's collected in a specific way. Can you talk a little bit more about the importance of that?
Well, that is just basic science where you cannot allow multiple variables to change and expect the same result. And there's a lot of things I know, and I know there are a lot of things I don't know. So things have to be done in exact manner, because if you do something different, I'm not quite sure how that's going to impact or skew the result. Maybe not at all, maybe greatly. I don't know. I don't want to know. I want things to be done very precisely, because I know when they're done this way, I'm going to get very accurate data. And I know it's a real burden on the patient to actually follow this protocol of just getting the blood drawn, and you got to shake it, and then you got to let it sit, and you got to get it in the freezer, and you got to put it on dry ice. There are a lot of things. The reason we do those things is because that gives me the most accurate reading on your physiology. So everything, all this pain of collecting the blood sample is done to just be the most accurate. And that includes, you have to do it in the morning. There are reasons behind all of these nitpicky little things. So the detailed processing is to ensure optimal accuracy in the results. I wish it were easier, but it's not. And that goes back to the molecule that we're looking at, the RNA. It's very labile. It changes, it degrades. I need to freeze it in time. When you get the PaxGene tubes, there's a solution already in the tubes. And what that solution does is it goes into your cells and it searches out the RNA and it locks it. It locks a molecule to it so it can't change, and it locks everything down in the cells. And then, but even though you've got this chemical fixing and locking of the RNA, what I want to look at, it still will change because it's in solution. Things in solution will always move. They'll always change. You cannot keep them static if they're in solution. And that's why this has to be frozen, and then it has to be frozen on dry ice to ship, to just lock everything and don't let anything move until I can get it into the lab and I can analyze it. And I think there's been a lesson in the CIRS community, and that maybe revolved around the measuring of complement 4, which is an important protein to measure for CIRS patients. And complement 4 is called a pre-protein, where it's a big protein, and then to activate it, you cut part of it off, and then the protein becomes active. So it's called a split product. And what happened was people were drawing this test, and once the blood is in the test tube, everything is still active. And that means this complement was being activated while it was just sitting in the test tube. And if you took this, and what people found, if you let this blood sample sit on the bench for 10 minutes and then drew another sample, now you have different results, because this one sample sat at room temperature for 10 minutes and this one was immediately frozen. And so you get wildly different results based on how you handle the samples and how you process the samples. We don't want that, because you don't know which result is real. So we have to be very precise when we're drawing this blood, and we have to be very precise in the lab. And sometimes there are things on the instructions where I say, don't put labels on the tubes, but you have to fill the tube up a certain amount or the result will skew. That's very important, because the blood has to mix in a certain ratio with the preservative. If you don't put enough blood in the tube, you don't have the proper ratio. If you put too much blood in the tube, you don't have the proper ratio. If you don't have the proper ratio, you won't get the proper results. You won't get accurate results. So everything is built around optimizing accuracy of the results.
I think a lot of people will really appreciate the explanation around the specificity, and I hope it helps to build some tolerance of, the instructions and to the frustration around it. But really, because there's a reason, and it's an expensive test and a highly helpful test. If you want to get the best results, that specificity is so important.
Yeah, and so the reason you can't put any other labels on the tube is because when you send that tube back to the lab, I'm going to weigh it to make sure you put the right amount of blood in there. And I can't have excess material on the tube, because that's going to throw off the weight. I always have to tear the labels off, and you have to get every little piece. If you put an extra label on there, I got to pick off every little piece, because I need to have an accurate weight of blood. So I know I'm going to make other calculations based on that weight. So there's always a reason for everything, and it's not apparent to a lot of people. But like I said, this has been many years in the making, and everything we do is for a reason. I'm always working to make things simpler, because I know that's a big hurdle for people just to get through this blood draw and get everything done properly. And so we're working on a couple things to make it a little bit simpler. And I think right now, we're also working with the U.S. Postal Service to be able to ship the sample back through the Postal Service instead of the commercial carriers, because there are a lot more post offices than FedExes or UPSs, and it's a lot cheaper to ship through the Postal Service. But we just had to go through and, because we're shipping blood, and you got to have all the proper safety mechanisms, and they want to know exactly, you can only ship so much dry ice, but hopefully that's going to bring down the cost.
That's great to hear. I think people will be appreciative to know that's being considered, and it takes time to refine the process of these types of things. I will say to people who are on the fence or, in preparation for doing this, and my heart goes out to you if you have a lot of brain fog when you're trying to do it. Just a real practical tip is, if you have somebody that can help you with the process, to double check what you're doing to help you locate where is that FedEx place that will actually ship dry ice, to help with the timing of the days and the schedule. You do need to consider when you're going to get the blood drawn and when you can ship it, so when it shows up at the lab at the right time. So there's a lot of different cognitive pieces that go into the planning for the test as well. But if you have anyone in your life that is not affected by CIRS and their brain's working, and in order to really bring them on board to help you problem solve those things so it goes as smoothly as possible, because you do want to get the most accurate results that you can. You touched on this a little bit when you said about receiving a sample and taking the labels off and things. I actually really appreciate hearing how you're handling our blood with such care. What happens once you receive the sample? On a simple level, can you walk us through just a process of what happens before we get the paper results?
Oh, yeah, I can walk you through that in my sleep because I do it a lot. So we get the sample into the lab. We first make sure everything is frozen, because if it's not frozen, we're not sure how long it's been thawed, what changes have been going on while it was thawed. So we ask that if it does come in thawed, we just get a new sample. It's not worth proceeding with the test because we can't be 100% sure the results are going to be accurate. That goes back to the very fragile nature of RNA and how quickly it can change in solution. So a lot of people out there, unfortunately, there's a delay in the shipping, or there's a problem and the sample comes in thawed, and we have to say, look, we don't want to process the sample. You have to just repeat the blood draw, and you don't have to pay for the test again, but you have to pay for the tubes, which is a hassle. So we get the samples in, and they are stored in a minus 80 degrees Celsius freezer until we're ready to start the processing. And when we begin the processing, we have to leave the tubes out and let them thaw. And once the tube is thawed, it has to be processed immediately. Once the tube sits out for 2 hours, then we start processing immediately. And part of that is, the first thing is we weigh the samples to see how much blood you actually got into the tube. And that's an important measurement we keep. And then from there, we have to spin down the white blood cells in the tube, and we wash those cells, and then we have to extract the RNA out of those cells, which involves a lot of chemical extractions and pipetting a lot of chemicals in the tubes and spinning the tubes down, and then running the tubes through a silicon filter, which will attract the RNA and let all the other junk wash out. And we wash the filters down, and then we have to elute the RNA off the filter. And that's the first step, called an extraction. That's an RNA extraction. So basically, someone sits in a chemical hood and adds chemicals and washes things and filters things and spins things. And you've got to do this for 3 hours straight. You're just washing and filtering and mixing chemicals. So you're sitting there for 3 hours straight processing this extraction. And then we measure how much RNA came out of your sample. And this is where the weight of the sample is important. People who have CIRS have what we've been calling hypometabolism, where the cells start going into kind of like a hibernation, and they stop producing chemicals that they need. This RNA is one of those compounds that they just produce less and less of it. The sicker the cell is, the less RNA it's producing. And so that's why we want to know how much RNA per milliliter of blood you produce. That's an important measurement, and that will define your hypometabolism. If your cells just aren't producing enough of this, we already know you're sick. We don't even have to run the test. I know you're sick just by how much RNA came out of your cells. That's one part of the process. So we extract the RNA from the blood. So that's a day's work, and that'll go into a freezer until we're ready to run the actual assay. And when we run the assay, we take a sample of your RNA and we mix it with some of the test reagents that we have. I won't go into the details of how the test works, but we work in a chemical hood and we mix a lot of reagents together, and we filter and we spin things down in a centrifuge, and over many hours of this process of extracting the RNA from your blood. And then we have a couple more processes similar, and I won't go through all the exact testing mechanisms, and it's probably not anything people are interested in. But in the end, we have to go through multiple different steps of processing the sample. And then we finally get to the point where we can read how many, your gene expression levels from your blood. takes many days to get work through this process. Each piece is pretty long. So multiple days of processing a single sample. And then we finally get to the end result. And that end result then has to go through a bioinformatic processing pipeline that I've built. And then we shoot those things into another program that was custom built for us to produce a report that the patient will then get.
When we talk about that price point of this test, and I hear you describing, the multiple hours, multiple days of getting the information that you need, to me, it seems fair.
Oh, I can tell you that. So I'm aware of two other tests that are similar, but not nearly as complex. And they range from $2,000 to $3,000.
Wow.
And they're both cancer tests. One's a prostate cancer test and one's a breast cancer test. And they use about 40 genes. They're looking at about 40 genes. We're looking at 180. And so we're a third of the price to a quarter of the price, and we're looking at 4 times as many things. So it's price-wise, the reagents that we use to process the test are very expensive. The machine that reads the final result, that one machine costs $250,000. So genomics is not cheap. I think we've talked about trying to get this test reimbursable by insurance. So maybe that'll help. It would also be nice if a bigger company bought us and did a little bit better than we can do. But I think if a bigger company bought us, the price of the test would go up to about $2,500.
Wow. Yeah, this test is really phenomenal, in my opinion, for those that are seeking further answers. And I know it's, as I mentioned already, it's impacted my life. Just want to bring up another place that this test can come in your treatment plan if you haven't done it in the beginning of your journey for whatever reason. After about a year of treatment, or if you feel like you hit a plateau, sometimes as a patient, you really need to see some progress. And sometimes symptoms take a long time to resolve, even though there are positive things happening in the body. So I decided to have my GENIE done again about a year later because I'm somebody that really needs to see the progress. And also, I was suspecting that I was having some sort of exposure again. I had a period in my recovery that went really well, and then I had a sharp decline again, which was unusual. And so I redid the test, and what was interesting is that it showed a huge change in my hypometabolism. So I had a lot of my cells playing possum and shutting down and really not working well, impacting my energy levels and my fatigue. And even though I wasn't back 100%, just being able to see my second GENIE test come back, which was a sea of blue the first time, showing that my cells weren't functioning well. And then to see in the second test a year later, almost all white, that they were now functioning as they should, that was a huge win. It was something that I really needed to see because I think CIRS patients need that sort of encouragement. They need to know what they're doing is actually working because all of this is so foreign. So this can be something that shows you progress over a year or show how things are shifting and changing. And that helps to keep the faith in the process and stay on the protocol. And it also showed that I was having continued actinose exposures and a different endotoxin exposure. And it made sense to me because I had felt like there was like a sewage smell or something coming and going. And so the test really confirmed that I needed to move. Again, I'm a renter. And so I had the ability then to finish up a lease and make a move. But I think the test can be used in a couple different ways. And I think just personally having something that can show really precise progress, and if you're having a continued ongoing exposure, is so valuable on the path.
Yeah, and I'll add to that something which is more anecdotal. Some people, when they develop CIRS, they will develop chemical sensitivities. You may now also be triggered not by microbial exposure, but by some type of chemical exposure, which is another layer of complexity.
Absolutely. A lot of people I hear talking about how they are impacted by different chemicals and things as well.
Yeah, once you kind of shake that immune system up and disorient it, which is kind of what's happening, it does things you just don't expect. And then you might develop other sensitivities from that.
On the GENIE, do we know, is there something that's upregulated or downregulated related to chemical sensitivity at this point?
We don't, but because we started running this test as a research use, which means if you take the test, we'd like to use it to understand things better. And there's always more work to do. And what we probably should do is go back and maybe send out an email to people who've had the GENIE saying, hey, did you have any chemical sensitivities that you're aware of? Or have you noticed anything unusual when you're around like a new carpet that maybe because it off gases formaldehyde and then you feel sick because you can't tolerate that level of formaldehyde or something. And then we can always go back and look, did these people have something on their GENIE that we could indicate they are prone to develop a chemical sensitivity from their CIRS?
Yeah, I could see that being really valuable.
And this goes to the CIRS community. It's just us. There's no one else. It's really a small group, and we got to work together.
Absolutely.
And the group really does. It's, everyone I think understands it, that there's just not a lot of help. There's not a lot of mainstream medicine that addresses this illness. And so we got to do it on our own.
Yeah, having said that, I'm curious what inspires you to continue to do this type of work and stay in the CIRS community.
Well, I guess the reward is hearing stories of people getting better. That's really what does it, I think. It's not really a money maker at this point, but it's a reward maker. It's just one of those intangibles that, when you've helped someone, that's a reward. It's rewarding. So hopefully we can continue to do that.
What are your deepest wishes for the CIRS community? Talking about the GENIE test. If you could have any wishes granted from the magic GENIE, what would they be for the CIRS community?
I hope we can just keep refining it and making it better. I hope more people with a background, maybe like mine in science, would become interested and add to the brain power. I think I'm probably a little outdated as a scientist. I'm not cutting edge anymore. You need some young blood. So I always look to see if there's anyone coming up that's interested that can keep pushing through to new ground. And that's where the answers are.
We're coming to the end of our time. Is there anything else that you'd like to share that we didn't cover in this conversation?
I think there's a group called CIRS, and I think they are trying to build new tools. I think we always need new tools. And I think the best way to understand and to treat this is to keep hearing stories and keep moving forward. And some of the doctors that have been working with Richie for a long time, I think hopefully they're expanding the resources for patients, along with surviving mold has always been the beacon in the darkness for CIRS. I'm just hopeful that we can get more interface and interaction with patients and success stories of what worked and what didn't work, and compiling maybe even a database of these things so that there's just more resources, a repository for everything, for all the knowledge that people have found in dealing with CIRS. So I just hope we can add more resources to the fight, because I think it's a very treatable illness, very treatable. I just don't know if it's an awareness issue, both in the medical community and in the general public. And I think things like your podcast and just getting information to people, however we can do it, and feedback, what works, what's not working, what don't people understand, how do we make it better, all of those things.
Thank you for bringing up all of those points. I think it is so important to get this information out there to the general public at large, just to increase awareness so that not everybody has to be able to diagnose it, but to be able to flag it and to send people along to the proper channels earlier would be so helpful. And while CIRS has a certain way that it unfolds for people, the way that people heal, there are certain details in that process that are different for everybody because we each have a different body and we're each in a different environment. And so what you're pointing to about sharing stories, the success stories, and including the parts that are real, the parts that like this did work or this didn't work, or I had to try this because of this circumstance, getting people to share their experiences in the early part and the middle part when they've overcome CIRS really is so important because every CIRS patient is slightly different. And so when these stories are shared and people, including doctors, can hear these slight differences because every doctor sort of sees different types of patients, it can help bring awareness to other things that they can pay attention to as well and bring that into their clientele. So sharing these stories and all of this information and helping it to be more digestible to the world at large, may that help so many people to come. I really do have so much gratitude for the work that you're doing and your commitment to the CIRS community and to this test and refining it. I really do think that this conversation will help people to feel a bit more confident in it and to understand the reasons behind it is the way it is. And many people respond to science, and I think science is important. But a lot of people respond also to stories like you're pointing to and feelings and conversations. So my hope with this podcast is that it will help to resonate with people in a different way that need to hear the message outside of the typical scientific presentation.
You're here.
Thank you so much for your time. Thank you for sharing all of this and for being a part of the CIRS community. And I really do feel so good knowing everybody's blood is in your hands.
Well, it was my pleasure talking with you, and thank you for doing what you're doing and bringing awareness.
May the awareness of CIRS spread far and wide, and may this test continue to be refined, and may it be of benefit to many. Thanks for being here today, Dr. Ryan. Appreciate it. Thank you for listening and for your kind attention. To keep in touch, follow the Heart of CIRS podcast on Instagram. You can visit melaniepensack.com/the Heart of CIRS to donate. Your generosity helps to keep this podcast growing. May the awareness of CIRS spread far and wide, helping to change millions of lives for the better.